Hi, everyone. Welcome to today's Hot Topic program, Human Origins Today. Today's program is Archaic Introgression in Modern Humans. What can it tell us about archaic humans and about ourselves? Featuring population geneticist Fernando Villanea. My name is Briana Pobiner, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray-haired woman in an orange sweater wearing headphones in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us or you've attended before, we're so glad to have you here. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of the Zoom interface. We're in a webinar format, so we can't see or hear you. As you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen. It looks like two speech bubbles, so that we can sort through as many questions as possible. The Q&A time really flies by. The Q&A box is also where we will share any relevant links during the program, so keep an eye out there. We'll start with an opening presentation by our speaker, Dr. Fernando Villanea, and then I'll join him here to take your questions. During the presentation, I may also write answers to some of your questions, at least any that I can answer, as will another member of our behind the scenes team, Dr. Ryan McRae. So now I'd like to go ahead and introduce our speaker. Dr. Fernando Villanea is a population geneticist specializing in human prehistory. He's interested in topics such as Neanderthal-human admixture and the effects of Neanderthal and other archaic genome variants inherited by people today. Dr. Villanea's current research focuses on the Neanderthal and Denisovan ancestry in American populations and how these archaic variants provided the genetic diversity for these populations to adapt to the novel environments found in the American continent. I'm really excited for this presentation. So now we'll turn it over to Dr. Villanea.
Hi, everyone, and thank you so much for having me. So let me share my screen and get going. All right, hopefully you can all see my screen right now.
Yep, we're good.
Yeah, thank you again for having me. Today I'm going to talk a lot about archaic introgression and what that means. So I'm going to start by talking a little bit about what is archaic introgression, because you're going to hear the term a lot. So let's think about all the people who are living on the world today, right? In different parts of the world. So we can imagine these meeples are people who live in different parts of the world today in the present time. We can also think of the ancestors of the people who are living today and how, when they go back in time, just connect into ancestral populations that are ancestral for multiple people. As we go farther back in time, all these ancestors are going to connect, all these branches are going to connect into one branch and ultimately one pool of ancestors for everyone who is alive today. So those are modern people and their ancestors. But we can imagine there might be some other branches. So in this little tree, this red branch, and that red branch doesn't quite make it all the way to the present time. So that is an archaic branch, that is an archaic population. It's a group of people who are related to us, but they became extinct at some point in the past, right? So we may think of them as Neanderthals or Denisovans, for example. But what's really key is before those branches became extinct, there were some episodes of admixture. So there is that dotted line that is horizontal that connects the red branch, the archaic branch, with maybe some, maybe all of the blue modern branches. And so that is archaic introgression, is the passing of genes from these extinct human populations into the ancestors of populations of today, of people who live today. Now, if we think about our genomes, what that means in terms of our genomes, so the part on the right of the screen. So the meeples here now are going to be different people who are alive today, and these strings of DNA are going to be their genomes. And we can pretend like black just means that this is all modern DNA, and then the little red segments, that is archaic introgression, right? Those are segments of the genomes that were inherited from maybe a Neanderthal, maybe a Denisovan, maybe some other archaic species. And some key aspects of archaic introgression is that if we look at multiple peoples today, we might find some people that may have no archaic introgression at all for their entire genome, or maybe a portion of the genome that we're looking at. We may find some people that are carrying some segments that are unique. That means that no one else has them in the sample we're looking at. We're going to find a lot of shared segments, right? So segments that are shared between different people. We're going to find some segments of the genome where no one carries any archaic introgression. So that is pretty typical of archaic introgression. What matters, is really important, is that if we then look at a bunch of different people and we collect the information in all these fragments, we are going to learn a lot. We can reconstruct a really large segment of the Neanderthal and the Denisovan genomes just from what is left on living people today. And we can use that information to learn about the past. So we can learn about how we're connected to these archaic branches. We can learn about the demography and the fate of these archaic peoples. And we can also learn about the present and the future. We can learn about what these elements mean for us, because these elements are not really inert. They actually have functions in our genomes. They affect our lives and they affect our health. And so these are the types of questions that I ask. How are these archaic elements relevant to us? How do they affect us? And before I talk about the archaic elements in us, I really want to go through a background of who are Neanderthals, who are Denisovans, just in case you've never been exposed to the literature. So I'll start talking about Neanderthals just because we know quite a lot about them. So Neanderthals were an archaic species of human. In the earliest reconstructions of Neanderthals, they were very ape-like. So this is actually one of my favorites right here. This is this drawing is called An Ancestor, the Man of 20,000 Years Ago from 1909. And what I like about it is that originally we thought as Neanderthals as some kind of like really distant relative who were very much not modern, right? They're very much not like us. We call them like cave dwellers. We thought they would slouch, knuckle draggers kind of thing, all these like epithets that are really derogatory. That's how I used to think of Neanderthals in the past. What's interesting is if you look at a modern reconstruction, the way we think about Neanderthals today, so the reconstruction at the bottom, that is a modern reconstruction of what a Neanderthal may have looked like. They look extremely modern, right? They look very much just like us, like they are just people. And part of this sort of revolution in the way we thought about Neanderthals, going from like thinking of them as being really primitive to thinking of them as being really modern, has a lot to do with DNA. Finding out that most of us carry some amount of Neanderthal ancestry means that if we call Neanderthal a knuckle dragger, that is a self-insult, because now we're insulting some part of ourselves. So we have to really, really rethink the way Neanderthals probably behaved and lived. We have a lot of different skeletal specimens, which means we have a lot of information about what they look like physically, but we also have a lot of cultural remains. So we know that they made tools that were really modern for the time, and they're just like really well specialized for their uses. We know they were extremely well adapted to a specific environment, which is the Eurasian environment, during some really cold episodes of time. We see that their tools were specifically designed for hunting megafauna, right? So for taking out these just really large mammals, sometimes probably really large some other things, which would have been really dangerous, but they were really, really specialized and very good at doing that. And maybe the part that has been most interesting, at least for me, is that we also have the genomes of 3 Neanderthal individuals that are really high coverage. One genome from an individual in Europe, the Villanea Neanderthal, and 2 genomes from Siberia, the Chagistria and the Altai Neanderthals. These are so good. These are so good coverage that they're comparable to any modern genome you can get from a person today. So these are extremely good quality. And that means that we have learned a lot about these Neanderthals from these genomes. We also know that by maybe 40,000 years ago, maybe 30,000 years ago at most, Neanderthals were extinct. All their populations have been in decline for some time. Their remains become really rare. And by 40,000 years ago, 30,000 years ago, we think they were gone. And this also coincides more or less with the time humans were, modern humans, that is, were expanding outside of the African continent on this migration that resulted in us spreading into every continent. So, and this could have been a coincidence, but what's important to remember is that the window of time in which modern humans and Neanderthals might have encountered each other might have been limited. I mean, still in the thousands of years, but it might have been somewhat limited. Now, this is a picture of the Strait of Gibraltar, because there is a really good Neanderthal site there. And it may have been one of the most recent ones. So these might have been the last Neanderthals who were living there in the European side and the Spain side of the strait. And something that I think about constantly that is very interesting is that if these were some of the last Neanderthals, that means that they might have never seen another human, another relative, throughout their entire lives. However, if you sit on the European side of the strait today at night and someone lights a bonfire on the African side, you can actually see that. The strait is not very long in that section. And that makes me think that these Neanderthals might have never seen another person during their lives, but they might have seen evidence of modern humans on the other side of the strait. And that makes me a little sad, maybe. But yeah. And then, of course, we have Denisovans. Denisovans are a completely different species than Neanderthals. They are as different, genomically at least, to Neanderthals than Neanderthals are to us. And the key aspect of Denisovans is that we know almost nothing about what they look like because we have no physical evidence for them. What we have is some fragmentary remains that were found in a cave. In fact, this is the same cave in Siberia from which the Altai Neanderthal is from. That includes some molars and some little pieces of pinky finger. Luckily, these were really well preserved, enough to yield DNA, and we can get a whole genome for that one Denisovan individual. And that genome is also really high quality, such that we can compare with modern genomes. That means that we know a lot about the genomics of Denisovans, but we know almost nothing about what they might have looked like physically. Some really clever approaches to maybe trying to figure out what they would look like include looking at Denisovan genes, compare them to modern human genes and Neanderthal genes, specifically genes that are relevant to bone growth in structure. And through that method, we have more or less guessed that if we were to see a Denisovan, they would have been robust. So much like Neanderthals, they would have been a little broader than modern people today. But that is probably as good as we're going to get when it comes to figuring out what they look like, unless we find some actual physical remains. There is one jaw from the Tibetan Plateau that has been linked to the Denisovans, so we know that they were present in that part of the world as well. But that is, as good as we have it for right now. But when it comes to the genomics of the Denisovans, we know a lot. And one of the things we figured out really early on is that Denisovans are mixed with modern humans multiple times. So we can see evidence of maybe multiple groups of Denisovans in different parts of the world who are encountering modern humans and they are mixing with them. There might have been as many as 3 different episodes of a mixture between Denisovans and modern humans. Now, I drew this little vignette so we can sort of place that in a context of geography and time. So let's think of the world and let's dial the clock back to 400,000 years ago. So at this time, we had anatomically modern humans. These are our direct ancestors evolving in the African continent. We have Neanderthals evolving somewhere in Eurasia, and we have Denisovans probably somewhere in Siberia, if not some other parts of the world as well. And at this point, these 3 groups are evolving independently. So we are accumulating a lot of our own unique genetic variants unique to each of these 3 groups. Now, there might have been some other groups that we don't quite know about yet, but we may see some evidence in our genomes, right? So maybe Homo ergaster in Africa, maybe some other divergent group of Denisovans, maybe Homo erectus, maybe Homo floresiensis, they might have also been present in these parts of the world and were evolving around these times. What's important is that about 60,000 years ago, modern humans started to disperse. First within the African continent, right, where we may have encountered some other humans, and then into Eurasia, where we definitely encountered some Neanderthals. And at some point during these exchanges, there was admixture, right? So genes were passed from one group to the other. So the little red and yellow dots, Those are archaic genome elements that modern human ancestors are carrying at this point. And as people keep moving, they were running into some other archaic groups, right? So now humans who were carrying Neanderthal ancestry also run into Denisovans. Admixture happens again. So now we have some modern humans that are carrying genome fragments from multiple of these archaic groups, right? And then by maybe 40,000 years ago, well, modern humans are the only group that are left. And so that means that all that is left of Neanderthals and Denisovans is in our own genomes, right? They still live on through their genes in our genomes. Now, this is a collection of every episode that's been described in the literature of contact between modern humans, which is the blue branches right here, present time being at the top, and then Neanderthals, which are the red branches, and Denisovans, which are the green branches, and maybe some of these other, what we call super archaic groups in yellow. And the point we try to make in this article here is that there have been so many of these events reported in the literature with evidence for just sort of independent points of admixture that it seems like passing down of genes, right, genetic admixture was not necessarily the exception. It was more or less the rule. Like every time any of these groups run into each other, they were probably admixing. And that is really challenging sort of this like biological species concept, right? Like if you remember, if you ever heard of the biological species concept, different species have to be reproductively isolated, right? They should not be able to reproduce with each other, but that is clearly not the case between modern humans, Denisovans, and Neanderthals. And so that makes it really difficult for us to think about if there were different species or they were just different populations and we are all one in the same species. But the end result of this long process of admixture between the different groups is the genetic ancestry we see today. So this is a really nice representation from Quantum Magazine in which they took populations around the world. So these are people who are alive today, and they quantified the percentage of archaic DNA for Neanderthals, Denisovans, and for super archaic populations. And then they painted them in a heat map where the cooler colors, that means that there's less archaic ancestry. The hotter colors, that means there's more archaic ancestry. And as you can see, everyone in these populations, they're all carrying some amount of archaic ancestry, right? We see a lot of Neanderthals specifically in Eurasia, but also in the Americas. We see Denisovan ancestry more or less mostly in East Asia and definitely in Southeast Asia. And then in Africa, we see some of the super archaic ancestry, and we also see Neanderthal ancestry as well. So most of us, if not all of us, are carrying some amount of archaic ancestry in our genomes. So today, I wanted to pivot into one specific aspect of my research, which is thinking about pharmacogenes. And I'm going to try to convince you that if we are thinking about studying archaic ancestry, maybe pharmacogenes is a really good place to start because they have a really good relationship with how we interact with our environments. So to give you a little bit of background, a pharmacogene is sort of a general term, it's an umbrella term for any part of the genome that allows you to metabolize chemical compounds, in particular things that are relevant to medicine. That's why the word pharma is in there. This is a big part of our mission for personalized medicine. Our goal one day is to be able to take your genome and then tailor medicine specifically for your needs. I drew a little vignette and to more or less what is the goal of studying pharmacogenes, right? So you can imagine that if you have a specific medical drug that you want to implement widely, it'll be important to then look at people's genomes and say, okay, well, this person is going to be okay with a full dose of this drug. You can imagine that some of these pharmacogenes sometimes are what we call a slow metabolizer. So they break down these drugs really slowly, which means a person would maybe need half the dose for that drug. On the other side of the spectrum, there are people who are hypermetabolizers. They process drugs really, really quickly. And that means that to get the effects of those drugs, you need to have double the dosage, for example. Or there might be people for whom these drugs just don't work. Their specific genetics means that we need to find an alternative. And so then you have to prescribe something completely different. And so that is sort of the goal of studying these pharmacogenes. The pharmacogenes that I was interested in for looking at archaic ancestry are part of a group called the cytochrome P450 genes or CYP450 genes. This is a really large family, includes hundreds of genes, but there's 11 of them that are the most important. So these 11 metabolize 75% of all medical drugs that, that we use today. And if you're thinking, I don't really understand how medical drugs are related to Neanderthals and Denisovans, because medical drugs are a recent invention. We've only had them for a couple hundred years. That is absolutely correct. But what's important to remember is that every medical compound that we use today is actually found in plants, right? In a plant today out there has all these chemical compounds that we use for drugs. And that means that these genes actually did not evolve to deal with medicine, they evolved to deal with plants that we're consuming. The reason it's so important for a gene to be able to cope with plant chemicals is because, well, plants have issues, right? They have problems. Plants can't really move, as we all know. That means that when plants get attacked by pathogens, when plants get eaten by insects, when they get eaten by mammals or birds, they can't really run away. So instead, what they do is they invest a lot of energy and resources in filling their tissues with these dangerous chemical compounds that are supposed to deter, us from eating them specifically, right? So plant tissues are just really, really rich and really dangerous chemicals. For some reason, though, humans are really, really good at coping with those chemical compounds. We have evolved specifically to consume a lot of plants that would be really dangerous to any other species. To give you some examples, a lot of the things that we eat are from the family Solanaceae, which is the nightshade family. The nightshade is definitely very poisonous to humans. Don't eat nightshade. But many, many things that we eat are in the same family, and they carry some of the same chemical compounds, right? So eggplants, potatoes, peppers, and tomatoes are all part of the Solanaceae family. Peppers, for example, really spicy peppers, the chemical that makes them spicy is called capsaicin. Capsaicin did not actually evolve to make your food taste good. Capsaicin evolved to keep you from eating food, right? It's supposed to keep mammals specifically from eating the peppers. Birds don't have the chemical receptor for capsaicin, so birds can eat peppers and they don't really feel the effects. Mammals are not supposed to be eating them, but humans love peppers, right? So we love eating capsaicin, and we have evolved to cope with that specific chemical. Another really good example is the family Anacardiaceae that includes the poison ivy and the poison oak, which are dangerous, right? You get really bad reactions if you touch them. But Anacardiaceae also includes things like the mango and cashews. And mangoes and cashews carry some of those same chemicals as well. There are people who are very sensitive to mango skins, for example, they can't really eat them without getting a reaction. Cashews, if you've never seen a cashew on a tree, this is what they look like in the middle picture right there. That's a cashew apple. They're edible, but they don't keep very well. So they don't really get exported. They grow in the tropics. But the cashew seed that we're familiar with that is right here, that is external. The cashew seed is actually toxic. You cannot eat the cashew seed if it's not being roasted, or you will get a toxic reaction, right? So we actually have to roast cashews and treat them before we can eat them. And even after they're roasted, if you eat too many cashews, you will get sick, right? So this is another example of plants pumping their tissues of chemicals to keep specific animals from eating them, but we humans have evolved to eat these things and do great with them. Some more examples include things that we know we can't feed our pets, for example. The reason we cannot feed our pets chocolate, the reason we cannot feed our pets coffee, and we cannot feed our pets grapes is because, theobromine in chocolate, caffeine in coffee, and the ethanol that you find in some grapes are actually really toxic. And to most other species, that means that they'll have a really bad day if they eat them, but not for humans. We have evolved specifically to eat these things. And the way we do it is through our pharmacogenes. Our pharmacogenes are really efficient at detoxifying a lot of these chemical compounds in the food we eat. And so when it comes to the archaic part of the talk, right, you can imagine that humans were evolving in Africa. We evolved to cope with very specific plants, right, that we detoxify really well. Neanderthals were evolving with some other plants. Denisovans were evolving with some really different plants because they're in different parts of the world. So they are evolving to cope with very specific toxins. But as humans, So as humans were only evolving in Africa, we would have probably had a hard time eating and detoxifying some of these other chemicals that we didn't evolve with. So this is a really interesting part of the research into Neanderthals and Denisovans. How were they coping with the specific things that they were eating? This is a really cool paper from a couple of years ago in which they did that, took some dental calculus from Neanderthals, and they used DNA to quantify what plants they were eating. I'm not going to spend a lot of time talking about the plants they were eating in general, but two things that caught my attention is that two of the specific things they found have medicinal uses. And one of them was the poplar tree. The poplar tree is actually not very nutritious. So you don't really eat it for nutrition, but you do eat it because it's rich in vitamin C, and it can help you treat some things like low-grade fevers, for example. So Neanderthals were eating some of these things that were, for their chemical compounds, not so much for their nutrition as well. So that makes it very, very interesting. So my collaborator, Katrina Klo, and I sort of took on looking at these pharmacogenes in Neanderthals and Denisovans. We were particularly interested in what variants they might have and how they relate to the variants we have. So this is why we are reporting this article that came out last month. This panel is the 11 most important CYP450 genes in humans. And these are the variants that we found in Neanderthals and Denisovans. I thought it was very interesting that Denisovans and Neanderthals tend to have different variants for the different pharmacogenes. But also, even within the Neanderthals, the 3 Neanderthal genomes that we have, there are some variants, right? Some of these Neanderthals are carrying things that are different from each other. We also label them based on their predicted metabolic efficiency. So if they are poor metabolizers, if they digest things really slow, they be somewhere in the red to orange to yellow. If they are normal metabolizers relative to living people, they're in green. If they are ultra rapid metabolizers, they be in blue. This is relative to the most common variants in modern humans. So we found that to be really interesting. Some other variants help them metabolize things really fast. Some of the variants make them metabolize things very slow, at least relative to most people today. We're also interested in seeing if any modern humans inherited some of these variants. And so this is another heat map. What this means is colors that are in the purple means that there is no archaic ancestry. And as you get closer to the orange, that means more archaic ancestry. These are different populations around the world, including Africa, East Asia, Europe, the Americas, South Asia, and Papua New Guinea. And these are the different genes that we're looking at in the previous panel as well. So whenever you see purple like this, that means that there is no archaic ancestry. No one really inherited CYP2C19 from these archaic humans. But a lot of people in Europe and a lot of people in the Americas and South Asia inherited, for example, CYP2C8 and CYP2C9. It's quite a lot of people carrying the Neanderthal variant of these two genes. And for CYP2J2, we also find that people in the America also carry the Neanderthal variant of this gene, as well as people in Europe and in East Asia. So that is very interesting. We inherited some of these variants that are specific to Neanderthals into Denisovans. And that makes a lot of sense, right? Because as modern humans were running into these new plants, they never evolved to cope with them. But what if, instead of having to wait for evolution to take its time to do that, we just do this, right? Yeah, we just take those variants from these archaic humans because they already evolved to cope with those plants. So we might as well just take advantage of that. Okay, and then finally, we were also thinking a little about natural selection. So in archaic genes, it's well known that natural selection has had a lot to work with, right? If we look at the human genome, so this circle is a representation of the human genome and all the 22 chromosomes and the X chromosome. In red and in blue, these are instances in which at least one person in the world is carrying Neanderthal and Denisovan ancestry. But as you can see, the circles are not completely painted red or blue, right? There are regions that are kept gray. And that means that no one who is alive today carries any archaic ancestry for those portions of the genome. And that is very common. We call these deserts of archaic introgression, and they can be quite large. Those are parts of the genome that are really rich in genes. What that means is that the archaic variants of these genes didn't really work really well in the modern human genome, and natural selection had to remove them. We've quantified the natural selection had to have been really strong and really good at removing these archaic variants in some cases. But in some other cases, the opposite happened. These archaic variants were really useful. And so natural selection would do the opposite. It actually make them more common. And so there's some very specific examples of genes for which the archaic variant are really helpful. And so we wanted to look at pharmacogenes in that lens, right? Is natural selection acting in these pharmacogenes? So for that, we looked at specific mutations in humans that we know are really dangerous, right? So these are mutations that if you carry that mutation, that pharmacogene probably doesn't work really well. You may have some issues and you may have to have some special care just because you can't metabolize some specific drugs. And out of all these mutations that are really dangerous in humans, we looked at them in the Neanderthals and Denisovans. We painted them red if we found them to be heterozygous, so they carry a single copy, and then blue if they have 2 copies of that mutation. That means they have both copies of the deleterious variant. And we found that a lot of these Neanderthals and also the Denisovan for one of the variants, they're carrying a lot of these really negative mutations, right? And they were like, in a modern human, these would be pathogenic. These would be dangerous. What we found is that even though the Neanderthals and the Denisovan carried them, no modern human today inherited them from Neanderthals or Denisovans. We don't see any of these specific mutations being contained within the haplotypes of these other variants that were inherited from Neanderthals and Denisovans. So I mean, that's the natural selection was actually really carefully removing these mutations once they made it into humans, which is what we expect. Natural selection works really hard to keep our genomes functioning. But we also were curious if natural selection had actually taken some of these variants and then made them more common because they're doing something useful. So we did something called PDS, which is a test for positive natural selection. We looked across all these genes in all modern human populations, and we found, at least so far, we found one example, the gene C2J2 in one specific population, which is in Peru. The Peruvians are overrepresented for that Neanderthal variant. That means that natural selection has actually taken that variant and has made it more common in Peruvians, probably because it's doing something really helpful in that specific environment. We don't actually know what that is yet, but we're working on it. And so just to finish up some conclusions, our genomes are a mosaic of one human, Neanderthal, and Denisovan genome elements, and that also includes the pharmacogenes. We know Neanderthals and Denisovans carry a diverse set of pharmacogene variants, and a lot of these were passed on to modern humans. But we also see natural selection doing a lot of work, right? So they're removing a lot of harmful mutations. And in at least one example, we see natural selection sort of using that variation that was added through Neanderthal and Denisovan admixture to work through adaptation, right? So we see one modern human population for which the archaic variant is seems to be doing something really useful. Okay, and so before I go, I would like to acknowledge all the people who were part of this project. This project was amazing, and I think the link has been provided for you if you want to check it out and learn a little more about pharmacogenes. And thank you so much for attending. And I am very happy to take any of your questions.
Awesome. Thank you for a fascinating presentation. And we do have a bunch of questions already, so we are going to jump into the Q&A. The first question that I'm going to ask is from Pat, who asks, is there any evidence of admixture between Neanderthals and Denisovans?
Oh, very good question. Yes, there is a ton of evidence for admixture between Neanderthals and Denisovans. It comes in all different forms, but I think the one that is like the most interesting is there is a genome for one ancient individual who is a direct descendant of a Neanderthal and a Denisovan. I believe it's a Neanderthal father and a Denisovan mother. The nickname for this individual is Denny1, and the genome is inclusive. Yeah, it was so lucky out of the few ancient genomes that we have to have one that is a direct descendant of the two. But we also see evidence of sort of really widespread admixture between the two groups. So Denisova K, the place where both the Denisovan genome and the Alpine Neanderthal genome comes from, there's evidence that they were taking turns inhabiting the same cave. And so at some points, they must have run into each other.
Well, exactly. We certainly have evidence that they did. So yeah. Here's another, I'll keep on the Denisovan theme for questions for the moment. Here's a question from AP who asks, the Denisovan variant for breathing at high altitudes, is that more common in people from Tibet? If it is, doesn't that mean natural selection has made it more common?
Yeah, no, that's a really good question. So yeah, the gene is called EPAS1. In Mono-Tibetans, about 86% of them carry the Denisovan variant, which is already pretty extraordinary. And there has been linked to important medical results, right? It helps them keep their blood thin as their hearts are working really hard to. Something that's very interesting too is that it's been estimated that the variant wasn't actually all that common during most of the evolutionary history of the people of Tibet until they moved into the really high altitude parts of Tibet. And there's evidence of that natural selection just taking over more recently than a mixture with Denisovans. So that the natural selection component of that happened in humans, not in the Denisovan, which I think is like
interesting.
super cool. It's one of the coolest results.
That is cool. Here's another question along those lines from Niels, who asks, have Neanderthal and Denisovan genes continued to mutate and evolve in modern humans? And is there any significant difference in how they behave compared to our modern human DNA?
Right. Okay. So two different questions. I'm going to start with the second one.
Okay.
The behavior in terms of the molecule is exactly the same, right? The information may be different, but the DNA aspect of that is exactly the same. However, the area is different, and right, of course, it has a lot of effects in humans. And we do see some amount of evolution. There is this other gene that I have been studying called MOC19, for which there is a Denisovan variant that is carried by people in the Americas. And it looks different because it has a duplication of a region of it, right? So like an entire portion of this gene has been duplicated in these American individuals. And that happened in the human side of the history, right? So the duplication doesn't come from the Denisovans. The duplication happens sometime later. So it's evidence that these genes are definitely still mutating. They're still changing. Wow.
Nice. I'm going to ask sort of an ancestry question also from AP, who says, given this information, isn't it more accurate to say that all of these groups are our ancestors, not just modern humans?
Yeah, that is fair. So I have the hardest time keeping sort of the words that I use consistent, even through my own talks, right? Because it'd be so much easier to say, oh, these are different species and they're not our ancestors, but clearly they are our ancestors. We're all carrying some portions of that ancestry. And so I have to pick and choose the words that I use through my talks just to convey information, but it definitely is not technically correct to say that only modern humans were our ancestors.
Yeah, that makes sense. And I think, we think of also sort of species ancestry versus sort of individuals that were part of a gene pool. And so I think it gets complicated. Here's a different sort of question from Camilla, who asks, is there any information about the skull found in China a few years ago? And I think she's referring to the Harbin skull.
Yeah, the homolongi skull. So as far as I know, there hasn't been any successful DNA amplification yet. And I know they're probably still trying. I'm really hoping they get some DNA. If I had to put money on it, I would guess that homolongi is probably a Denisovan. Definitely their skull is really robust. I've been told if you hold it, it's just as gigantic. Which it goes along the expectation we have for Denisovans being robust as well. But unless we get DNA from it, I think it's going to be really difficult to tie it specifically to Denisovans.
Yeah.
Interesting fact is that Denisovans don't have a species name because we don't have a holotype. If the Harvey skull happens to be a Denisovan, that means the Denisovans will be homolongi.
Oh, that's true. So they won't be something like Homo denisova or something like that.
Right.
Yeah. So here's a question from Rick who asks, how might we determine or reject the possibility that genes shared by Homo sapiens, Neanderthals, and Denisovans could have derived from a super archaic or several super archaic species?
Right. Yeah, that has been some of the more interesting, I think, research points lately, is looking at this super archaic ancestry. So we definitely have evidence for super archaic ancestry. Um, the reason we know it's super archaic is because we can look at something called the expected divergence between
Mhm.
portions of the genome, right? So if we look at one portion of the genome in all modern humans, we're going to see that most people have some expected number of differences that are consistent with mutation in time, right? So the mutation rate, given the time humans have evolved, should lead to some number of mutations in that portion of the genome. But then a few people will be carrying a segment that just has way too many of these mutations, right? More than expected. And so because we know what the expectation is, we can say this is outside of that expectation. So this must have come from a branch that is much farther divergent than the majority of the genome. Because these expectations are tied to time and a mutation rate, and we know the mutation rate, we can also say this divergence looks consistent with 500,000 years, which happens to be the Neanderthal and Denisovan, the timing for the branching.
Mhm.
But some of these fragments are much more divergent than that, like twice as much, 1 million years, 2 million years is the
Mm.
expectation. Those we have to consider as being super archaic in ancestry because they don't really match the expectation for Neanderthals for Denisovans.
Okay. Thank you for explaining that. Here's a question from Steve who asks, do you see gene mutations responsible for causing specific diseases in Neanderthals or Denisovans?
Oof, what a question. Um, so we don't see anything specifically tied to a particular disease, but that has less to do with Denisovans and Neanderthals not suffering from these diseases, and more to do with the fact that most diseases that we know of are extremely complex when it
Mm.
comes to the genetics. So there's very, very, very few of what we call Mendelian effects, in which one gene corresponds to one particular phenotype. Instead, what we see
Mm.
is that most of these medical conditions, there's hundreds of genes that are responsible for them, the interaction between these hundreds of genes. And so that makes it very difficult to even make a direct connection in humans, for which we have living people to compare
Mm.
notes. It makes it almost impossible to do it
Mm.
in any of these ancient genomes.
Yeah, that's fair. Walter asks, is it possible to correlate the emergence of new bacterial and viral infectious diseases in humans with a varying diet caused by human migration?
Hmm. So let me think about that for a second. It's interesting because a lot of the archaic introgression genes that we see that are selected for in humans are related to the
Hmm.
immune system. And that makes a lot of
Hmm.
sense, right? Because yeah, modern humans are moving into all new environments, and in addition to new foods, they're also running into new pathogens.
Hmm.
Neanderthals and Denisovans certainly had their own pathogens that were probably different than ours.
Hmm.
And that means that there was some interactions there with our immune systems running into
Hmm.
these new diseases. I wish I had a more concrete answer, though.
Yeah, there's a lot of things we don't have concrete answers for, but I think it's helpful to think through how we might find those answers. So yeah. Here's another food related question from SS who says, I find the idea that we can eat things other animals can't very interesting. How do you think this intersects with cooking and the preparation of food? I remember there was evidence of something like this with Neanderthals in the Shanidar caves. Could this have changed how well we metabolized technically toxic compounds?
Yeah, no, that's really really good to start thinking about that as well, right? How culture interacts with these biological quantities. I think I was mentioning cashew seeds before, right? So you have to roast those before you eat them, otherwise you're gonna get sick. So that is 100% interaction of cooking with something that we eat all the time, and we eat roasted cashew seeds all the time. And similarly, cooking definitely can help break down a lot of these chemical compounds. A lot of them are not thermally like stable. And so you can get around eating a lot of these toxic things through cooking. I love to think about who might have been the first person who made the experiment of eating something raw and then cooking it. I imagine there were a lot of these experiments happening through human evolution. But yeah, no, cooking is definitely an integral part of this story as well.
So we have a couple of other questions from different perspectives. Here's a question from Sudi who says, very interesting. How close are scientists to using your findings in developing treatments?
Okay, well, first of all, I'm going to plug in my my colleague Katrina Kloh, because she actually is in the School of Medicine. She deals with the medical part of the pharmacogenes. But that is 100% their mission. And we already, but like medical doctors already implement some of these tests. I believe for morphine, for example, or some other of these like codeine-derived anesthetics, they looked at your specific variants of the CYP450 genes that you carry before they prescribe your dosage. Because it's really dangerous, right? Like if you are a slow metabolizer, if you break down some of these codeine-based drugs really, really slowly, you can overdose from what would be a regular dose to anyone else. So they already do testing for a lot of these for very extreme things, right? So extreme situations. I believe for asthma medication, they also test specifically if it's going to be effective for you or not. And some other more curious things, I think, I want to say 23andMe does some of the CYP450 testing. They'll tell you if you're a caffeine hypermetabolizer, which is CYP1A2, which I am actually, which explains why I have to drink like 6 cups of coffee a day. So if that's something you have done, you should go and look at your results. They'll have a couple of these pharmacogenes in there.
Oh, that's pretty cool. I have a question from our behind the scenes team, actually, and then I'll get back to some audience questions. So is there a way to differentiate whether a pharmacogene is being selected for due to its beneficial effects in a population or just being overexpressed due to a lack of genetic diversity, something like a founder effect?
Okay, really good question that I have to deal with all the time in reviews. So the test that we use for this PBS, one of the benefits of it is it generates the statistical distribution for it to see if something is actually like statistically significant or not. It's based on the entire genome. Founder effects, for example, they affect your entire genome, right? A founder effects doesn't care if something is a gene or if it's doing something important. Founder effects just affects your entire genome, but you'll see sort of the extremes of these distributions for variants across the entire genome. So we compare across the entire genome. And we say, for example, for the C2J2 variant, we found that is overrepresented even relative to all these other variants in the genome that are overrepresented because of the founder effect. So yeah, it's a great question from what I'm assuming is a reviewer in there.
Maybe in the future, I don't know.
Right.
I'm going to ask another a question from our behind the scenes team who said, maybe you mentioned anatomically modern humans at 400,000 years ago.
I believe I did.
Okay, because I think that the date that we have often most seen is 300,000 years ago. So it was in a figure. And so we were just sort of curious about that.
Yeah, I mean, I am not an expert in the paleo side of things.
Fair enough.
So it's really hard to say. And I leave that to the bone people to define, to draw the line for anatomically modern humans. But what I do know, just from looking at genomes, is that by 400,000 years ago, our gene pool was different from the Neanderthal and the Denisovan gene pool.
Okay. So it may have been sort of
They're
like that
evolving,
ancestral population of modern humans.
right?
Okay, great. Thank you. So here's a question from Augustin, who asks, is it possible to trace back to chimpanzees given our 98% similarity? And I think that question came in as we were talking about that super archaic potential influence.
Right.
And then he followed up and said chimpanzee CYP genes.
Right. So yes, and there are a lot of sequence chimpanzees that we looked at actually for these 450 genes as a comparison. When it comes to the super archaic ancestry, I mean, I totally believe that we could have some chimpanzee donations along the way as well. But once again, because we can calculate the expectation given time, and we have something like a 6 million year window for the divergence of chimpanzees in humans. Those segments don't appear quite that diverse. They're definitely not 6 million years diverse. They're more like in the 1 million to 2 million year diverse.
Okay, that's helpful. I think I only see one more question, so we'll get to this question and we'll see if any else come in. Otherwise, we will wrap up. And so Sylvia asks, as an aside question, what's the picture on your shirt?
Oh, it's a kiwi.
Oh, very nice. Excellent. Great. Thank you. All right. So I am really grateful for you, Dr. Villanea, for your wonderful presentation. And I think we will go ahead and wrap up. So this concludes today's virtual program. Please join me in thanking Fernando for sharing his work with us. I'd also like to give special thanks to those who made this program possible, to our behind the scenes team who helped sort through your questions, including Dr. Ryan McRae. I'm just looking real quick to see. Great, perfect. If there were any more questions, who works with us at the Human Origins Program, to our donors, volunteers, and viewers like you. And finally, to all our partners who help us reach, educate, and empower millions of people around the world today and every day. Thank you. And you're getting some thanks also coming into the Q&A. So next month on February 15th at 11:30 a.m. Eastern Time, our hot topic presentation will be What Chimpanzees Can Tell Us About Human Evolution with Dr. John Matani, who is a primatologist. So that will be from a behavioral perspective. We've put a link in the Q&A where you can find information about our upcoming programs and how to sign up for the museum's weekly e-newsletter. That's the best way to stay informed on upcoming programs and learn more about the museum's research and exhibitions. After this webinar ends, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond to that survey. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. Again, thanks to the participants. Thank you to Dr. Villanea for your presentation and to you, the audience. We'll see you next month.